Uh, Welcome back to the class. So, in this second chapter that we started in our last class, we learnt about what is the male gametophyte in a plant system. We saw the structure of anther, we saw the structure of microspore, we saw how microsporogenesis happens, and we saw that because we're talking about the sexual reproduction in the case of plants, this gamete, the male gamete has to fuse with the female gamete, right? We saw about the pollen grains and the use of pollen as well in our last few classes, right? Now in this class, let us try to focus on the female gametophyte and how the female gamete is produced, uh, say, in a flower. So we've already seen the structure of a flower. We know that the structure of a flower has the ability to produce both the gametes or either of the gametes, right? And we have seen the structure of the male gametophyte. Now we will study about the female gametophyte, what makes the female gamete, and what exactly are the structures that are involved in the making of a female gamete, right? So to start off, the innermost whorl in a flower is what is called as the female reproductive part. Okay, now that female reproductive part is what we call as the gynoecium of a flower. uh, in almost all the flowers, if there are both the sexes present together, you see that the gynoecium is the innermost whorl. Otherwise, you see in a few cases where there can be many, I'll tell you the different parts of this gynoecium in some time. If there are many carpels and all of them, you see that they can be fused, they can be different, but whatever said and done, they would always be towards the center of the flower, right? So you understand why this gynoecium has to be in the middle part of the flower in the first place. Gynoecium is the place where actually the seed formation takes place, and if by chance it is somewhere in the periphery of the flower, you see that it can get damaged, and hence that should just not happen. Therefore, the female gamete is kind of embedded, you can say, in the flower, not like the pollen, which is kind of exposed, where the female gamete is present, kind of well protected. Okay, now this gynoecium might have a single pistil. Okay, if it has a single pistil, we call it as monocarpellary. We call that as the monocarpellary. Uh, but not all of them can be monocarpellary. Like a typical flower we saw had only one pistil raising, right? So you can call that as the monocarpellary. But many times we see that it's not monocarpellary. There are many pistils present. So there can be numerous pistils that can be present, and when there are numerous pistils, you call that as the multicarpellary flower. And I'm sure you still remember what you had learnt last year. We said that suppose there are more than uh, one pistil, they can be fused at the base. We call that as the syncarpus, which means to say that they are fused, or they can be free, and that we call it as the apocarpus, right? So the pistils can be fused, the pistils can be separate, there can be one pistil, there can be many pistils. Now all this diversity is required because depending on where the plant is growing, depending on how the pollination should take place, depending on how the seed should germinate, you have varieties of gynoecium that's seen in nature, right? Now whether we are talking about a monocarpellus flower or a multicarpellus flower, syncarpus or apocarpus, you see that every pistil has three important parts. Okay, now they are, so we had this thalamus here. No, so let me just say that this is the thalamus. Now in the thalamus you see that there is one small region, which acts like which has a ball like structure at the base. Then this is elongated, and then you also see that at the tip it would kind of get branched. It would get branched like this, and this is how our gynoecium looks like. Now this gynoecium which we are talking here, you see, has the exposed part which we call as the stigma. This exposed part is what we call as the stigma. Very important. You can, if you, in fact, if you see it under the microscope, you see a very small, thin, hair-like structures here, which would kind of help to trap the pollen grains if at all it falls on the stigma. Uh, Also, you see that there are some kind of sugary substances which kind of helps in the attachment of the pollen grains, right? Then comes the most elongated region of the gynoecium, and this is what we call as style. Style basically in biology means that it is a long, very slender tubule. Okay, it's a very small tube. Now, it's a very important part of the flower because it is through this that the pollen grain actually gets matured, right? We said the pollen actually is giving rise to gametes. It's not gametes by itself. Now, this style provides an environment for the pollen to kind of start dividing. Okay, so the style is very important, and then at the base we have this region which is called as the ovary. So we have these three of them, very important part of our gynoecium. Now we know what stigma does. It helps as a platform for the pollen grains to come and attach itself. Then they would all move towards the style, and then they would reach the ovary. Now the ovary is not just like a ball-like structure that we have written here. Okay, it is quite complicated structure because inside the small region which is called as the ovary, we see a small cavity which is called as the locule. Okay, I'll be writing the structure soon. And then there is a placenta, and we know what placenta is. Placenta is basically the region which helps in the development of the seed. Okay, and from the placenta you see some spores, like not the microspores, we have the big spores called as the megaspores that comes, and from that we get the ovule, right? And that is what is very important, as that gives rise to seed later after fertilization, right? So how does a ovary look like? Now if I just look at the ovary, you see that it is made up of many different types of layers. uh, so let me just write one of them here. uh, so there would be embryo sac, and on this embryo sac you would see one covering, and that covering is something which kind of protects the whole of the embryo, right? And then you also see there is one more layer. This is called as the first layer of the seed. We call it as an inner integument. Now above this you have one more layer which we call as the outer integument. This becomes much narrower. You can write it like this. So you have the outer integument, right? And this outer integument which we have gives rise to a small pore. Okay, it gives rise to a small pore, and then you have the region which kind of covers, encloses all this, right? So this would be the structure here, right? So you see that this outer region, innermost region, is what we call as the embryo sac, and above that we have a region which we call as the nucellus. Nucellus is a layer which protects the embryo sac. Then we have the inner layer of the seed, which we call as the inner, it's called as an integument. Integument basically means membrane, that is all it means. Okay, so we call inner integument, and then you have the outer integument, right? So we have the outer integument, and there is the small region through which a small pore, I can say, pore, which from where the pollen would enter and kind of fertilize all this. We call that as a micropyle, right? So it is called as a micropyle. So you see that this whole of the embryo looks like this, but if you just keep on going inside, you see that there are so many layers around, right? And most of the formation of the seed and development of the embryo happens at this part of the embryo, and there is one part which is actually not playing an important role, right? Now that pole is what we call as the chalazal pole. We call that as the chalazal pole. So basically, if you look at this, looks quite complicated, right? But actually it's not so. Maybe when when you have practical classes, you will be given seeds where you can actually see the different micropiles, integuments, and all that. You can take seeds and the flowers, and you can very well get to know how all this is very important, right? So here in the embryo sac, there is placenta, which gives rise to the ovules. Okay. Now comes one more important aspect. We have ovules. It can be one or it can be many, right? If it has one, you just have one seed, say like a mango fruit. You have seen that there is only one seed there, one large seed. Or sometimes you would have seen so many seeds present, say like watermelon, right? Watermelon has so many seeds, so that they have multiple ovaries. Okay, so now let us talk about this ovule in a bit detail. Okay, now this ovule, is a part of our ovary, and the whole of the ovule is very important because it has the ability to form seed. Right? Now this ovule is what we call as the female gametophyte, or many times we also call this as the megasporangium. It's called as the megasporangium. Now the megasporangium is nothing but the ovule. Megasporangium is nothing but the ovule. Now this whole of the ovule is attached to the ovary through a very small minute thread-like structure, which is called as the placenta. Okay, but the placenta is a term which we generally use it for animals, whereas in the case of plants, it is attached to the ovary through the small pipe-like structure that is here, which we call as the funicle. Okay, we call this region as the funicle. Now you see that there is this funicle which is having two protective membranes, which are the outer membrane and the inner membrane. Now we don't call them as membrane mainly because these are completely protective in structure. We call them as integuments. Okay, now this integuments is covering throughout the important embryo sac, except in one small region which we call as the pore. Now this pore is so small, so I have to call it as micropore. Now since the pollen enters here, we call it as pile. So the name of this pore becomes micropyle. Okay. Now this micropyle is that region through where the pollen grain would enter and fertilizes the ovule. Okay. So you see that after the membranes are there, we have the nucellus. Nucellus is one of the most important part of the seed. The reason why that is important is because this has abundant reserve food materials, very much like our amniotic fluid, which has very good food reserve materials for the growing embryo, right? So here the whole of nucellin is very important, mainly because this helps in uh, kind of taking up the nutrients and many more, right? So comes one more important thing here. Now, as you go down the nucellus, there is this embryo sac. This embryo sac is nothing but the female gametophyte. Okay. Generally, you will have one single embryo, and that is actually formed by meiosis. Okay. Now, the whole of reduction division that happens inside this megasporangium is what we call as megasporogenesis. It is what we call as the megasporogenesis. Okay, now what is mega? Like just like how we studied microsporogenesis, megasporogenesis is also a process where there is one megaspore mother cell. Okay, uh, so we have one megaspore mother cell, and this mother cell would finally give rise to one megaspore. Okay. Okay, I will not tell one megaspore because you will very soon realize that it might be one or it might be different. Okay, so we have one single megaspore mother cell in one ovule, and in the micropylar region, you see that there is some kind of division that would happen, and this would give rise to megaspore. So there is one mother which would undergo reduction division. So how many megaspores are actually formed? So you see that if there is one MMC, you get four megaspores. Okay, so this is how generally it would look like. Okay, now in most of the flowering plants, you see that the megaspores is uh, very characteristic. That is because there are four megaspores, right? So let me just write like this. So there are four megaspores. Now there is some kind of activity that would go on in a plant, and the plant decides to keep only one gametophyte functional. Okay, so there is only one of them which is functional, and the other three would undergo a process of degeneration. Okay, they would just undergo degenerate, and this female functional one megaspore would finally give rise to embryo sac. Okay, so this finally gives rise to embryo sac. Okay, now this is something which is very commonly seen in most of the flowering plants, and since there is only one of them of the four which is a functional gametophyte and which will give rise to a functional embryo sac, we call this whole development process of the megasporogenesis as monosporic. Mono because there is only one of them, one cell which is behaving like a spore. Okay, uh, so this one is monosporic, will always be haploid in nature. Okay, Uh, so this is how you see megasporogenesis would be actually formed. So I hope you do not have any doubts with this, right? Okay, now let us just continue further and see how the formation of the embryo sac actually happens. Okay, uh, we have a functional, I would just erase this, even this. So there is only one functional megaspore, right? So let me just call this one functional megaspore. Now this functional megaspore would undergo a mitotic division, okay, and it gives rise to two daughter cells. So let me just write that it is a mitosis that is happening. So there is a mitotic division that would happen. Now, whenever there is a mitosis that would happen, what we generally see is that there has to be two cells, right? But in this particular case, the mitosis happens, but the cell division does not happen, right? So you have one single cell. Let us call this as the functional cell. Now here there is mitosis. Uh, Let me say there is one nucleus here. Now here there would be, after mitosis, two nuclei. Okay. Please do remember that this division that is happening is mitosis, right? Now, when the cell would have actually divided here, there would be two cells, but that does not happen. After meiosis, there is one mitosis that happens. Now, in one single cell, there are two nuclei. Now, I'm sure we would have discussed in many of our live classes that this kind of scenario is kind of safe, but not always very safe, right? This can happen only under the presence of certain proteins which kind of uh, help them to stay together like this. But even then, you see that there is some kind of repulsion that would happen, and then the nucleus which are formed, the two of them, would actually move to the opposite poles. Okay, so if there is only one nucleus, it is always in the center, but we're talking about the plants here, so the nucleus itself is smaller compared to the vacuole. In spite of that, you see that here there is formation of two nuclei and they are moving to the opposite poles, right? Now there is one more important stage that would happen. This nucleus, please do understand that mitosis generally gives rise to two cells of the same ploidy level. Here there is mitosis happening, but there is no cell division that is happening. Now this stage cell would undergo mitosis again, okay, and then there would be a stage where there are four nuclei. This one would divide twice, this one would divide twice. So there is a four nucleate stage. Now from the four nucleate stage, you see that it comes to one more stage where there is 8 nucleate stage. Right? You're getting what I'm telling you. So basically there are 8 nucleate stage. Now this is something which is very important because in one single cell there are 8 nuclei, okay. There is no cell wall, there is no cell membrane, and therefore there is basically one single functional megaspore which gave rise to a huge cell, okay, huge cell where there are 8 nuclei present. Okay, now after the 8 nuclei stage is reached, then you see that there is formation of cell walls. Okay, there is formation of cell walls in this 8 nucleate stage, and then what you get is a structure very much similar to our female gametophyte, or many times which we also call it as the embryo sac. Okay, you notice that uh, around 6 out of the 8 nuclei, they are completely surrounded by the cell walls, and only 2 of them are basically organized into cells. Okay, uh, so basically the whole of, after division, after the whole of the division, how it would look like. It would, this 8 nucleate stage would very much look like a stage of the embryo sac. So let me just write my embryo sac here. Now this embryo sac at the chelazal end would have 3 nuclei. Okay, it would have, so I will write the 3 nuclei that are present there, and then in the other end, which is the micropylar end, you see that there is formation of another 1, 2 and 3 cells present together. Okay, and somewhere in the center you will have the 2 nuclei that are present. Okay, now this is a very important concept because you will realize the importance when we start talking about the double fertilization. So just for now, you can remember that one functional megaspore cell would undergo mitosis, nuclear mitosis, that would be a better term to use. It undergoes a series of nuclear mitosis and would finally give rise to 8 nuclei. Now all the 8 nuclei which are there would kind of move towards the poles, such that 3 of them would come to the chelazal pole. These 3 would come to the chelazal pole, 2 of them would remain in the center, and the other 3 would move to the micropylar region. I hope this part is clear, right? Now if I just look at all this, I would erase this part of the diagram now. Suppose I have, let me just write only the embryo sac now, only the embryo sac. So we have the chelazal end here. I would not write that diagram, I would write the 8 nuclei. So there would be 3 nuclei, right? 3 nuclei which comes to this part of our female gametophyte. We call this as the antipodals. Okay, we call this as the antipodals. You know what is antipodal? Podal is what? Podal is something which helps to move. So this actually does not allow it to move. Basically, it's that. Okay, so we have antipodals, or 3 nuclei which comes to the chelazal region. So these are the 3 nuclei. Then there are 2 of them in the center. So let me just write them here. Now these are called as the polar nuclei. Okay, we call them as the polar. These are called as the polar nuclei. See, the polar nuclei is very important. Why? Because this is the one which is very important for kind of a well-being of the entire embryo sac. Very soon you will realize which nuclei will fuse with the pollen grain and all that very soon. Okay, and then comes another 3 nuclei which goes to this part of the micropylar region, where there are some kind of sac-like structures, okay, bag-like structures, which we call them as the synergids. Okay, we call them as the synergids, and the 3 plus 2, 5 is over. Another 3 would be in the synergids, and there is also egg present in one of these synergids. Okay, so there is one of this sac which behaves like the egg. Okay, but why does this actually? Why is this nuclear mitosis happens? And what is the role of all these 8 different nuclei? Why is there so many nuclei? and which one is actually going to give us the seed is what we are going to study when we learn about fertilization. So this is about the female gametophyte. Okay, I hope you do not have any doubts. Okay, uh, no doubts, right? uh, I have one very interesting uh, you know uh, thing about the female gametophyte which I would like to tell you, and that is in the whole of the reproductive system of any higher plant or an higher animal, you see the plant gametophyte is a very special different type of cell, mainly because the embryo sac, which we call as the female gametophyte, is the only one type of cell which has 8 nuclei present in it, and basically that entire sac derived from one single functional megaspore has different cells present. There are basically 7 different types of cells, 7 cells, not different types, present inside this embryo sac. For example, this each one of them would give rise to 3 cells which we call as antipodals, then we have the synergids, and then we have the polar cell. So basically the whole of the embryo sac is 8 nuclei and 7 celled structure, right? However, the importance of all this we will be studying in our next class. Right now, once we have the female gametophyte producing all these structures, the male gametophyte which has already produced the pollen grain and is ready to be dispersed, it's already desiccated and dehydrated, and now it's kind of matured. The next important aspect when of the whole of the sexual reproduction in flowering plants is the movement of the pollen grain towards this female gametophyte, and that process happens by a different mechanism which is a characteristic of the flowering plants. That process is what we call as the pollination. So over to understanding what pollination is now. So we have studied the male gametophyte, we have studied the female gametophyte. Now we will study about the next most important aspect of sexual reproduction in flowering plants, which is called as the pollination. Uh, Please do remember that plants themselves are non-motile, right? The gametes also are non-motile, but now we are talking about sexual reproduction where there has to be fusion of these two gametes. Now, how does that happen? Now, that happens by a process which we call as the pollination. Okay, basically to define, I would say it is nothing but the transfer of the pollen grains. Okay, please do remember the pollen grains which are not attached to the anther, they are already desiccated, they are dehydrated, and they are almost vulnerable to come out of the anther. Okay, so that kind of pollen grains which are uh, coming out of the anther, if it falls on the stigma, you call that entire process as pollination. Okay, the best part about pollination is that every organism, every plant, every animal, every insect which feeds on flowers, they have their own modifications, they have their own adaptations to see that pollination actually happens. Okay, and pollination is successful. Uh, so there are many kinds of pollination that can happen, and you see the bright colors in a flower, or the flowers which are beautifully smelling, or they might be sweet. So all these are kind of aids which help in pollination, right? Now, depending on the source of pollen, we have pollination which is divided into 3 broad categories. Okay, so the first category of pollination is what we call as autogamy. Now, auto means what? Auto means same, similar, happens by itself. So auto is a pollination where there is only one single flower, and pollination happens within the same anther of one flower to the same stigma of the other flower. So in a normal flower which opens, it exposes the anthers, it exposes the stigma. You see that autogamy can be rare because the anther can fly out sometimes. But autogamy, is generally very commonly seen in those flowers which are always closed. Also, please do remember that the maturity of the male gametophyte and the female gametophyte should be perfect. There has to be something that we call as the synchrony, right? So there has to be synchrony in the release of pollen, in the development of female gametophyte, in the development of the stigma, which all this leads to the development of stigma, and all this should be in some kind of synchrony that happens. So you see that this kind of autogamy can happen only in those plants where the flowers are basically small and the pollen grain and the stigma are very close to each other. Okay, I'm sure you would have seen this flowers like oxalis. Oxalis is that huisopu which grows in our houses. Okay, now this oxalis or comminalia, you see that they produce two types of flowers. Okay, one which are similar to flowers of other species, where the flowers just bloom, there is anthers, there is stigma, style and everything, and they are exposed. Okay, so such flowers which kind of open, we call them as chasmogamous. Chasmogamous are basically the flowers which open up, okay, which bloom. And when they bloom, what would happen? They are exposing the male and the female gametophyte. Whereas many times you see that they would be closed also. We call such flowers as the cleistogamous. Cleisto means closed. So cleistogamous basically means that they are closed all the time and they do not open. Now, if by chance the uh, anthers and the stigma are very close to each other, you see that autogamy is possible. Uh, Whereas if they are too far away, then autogamy does not happen. Also, in cleistogamous, autogamy is favored because they are always together, they are closed, so they have to undergo autogamous only. Whereas in chasmogamous, it can be different. Okay, so this is the first kind of pollination, which is called as the autogamous pollination. There is second type of pollination which we call as geitonogamy. Okay, now basically geitonogamy basically means that it's a cross pollination. Okay, what exactly happens here is that you have two flowers. They might have both the sexes present in them or there might not be, but whatever you see that there are two flowers, the anther of one flower goes and falls into the other flower. Okay, now when we are talking about geitonogamy, it is always possible by the help of some kind of pollinating agent. Something should carry the male pollen grain to the female gametophyte, right? So it can be air, wind, which we will see later. So if there is the flower, which is there are two flowers involved, we call it as the geitonogamy. But now there is one condition here. In geitonogamy, the flowers has to be from the same plant. Okay, it has to be from the same plant. Just to give you one example here, look at the beauty of our nature. A plant generally, if it flowers, if it has to flower many, if it has the ability to flower many flowers, you see that all the flowers would bloom almost at the same time within a short period, say today, tomorrow, day after, or within 3-4 days. Uh, they have something that we call as season. So all this will actually help in the maintenance of increased geitonogamy and increase that same kind of seeds formed. So they will be exactly genetically same as that of the parents, because the seed formation happens between the male of the same plant and the female of the same plant. However, at least their same species can continue, right? And in fact, that is the reason why most of the flowers bloom at the same time in a plant. So that is called as the geitonogamy. And then we have the third type of pollination, which we call as the xenogamy. Okay, now xenogamy is completely different. You know what is a xenograft? Xeno means completely different. So one plant from one, one flower from one plant, another flower from another plant. So there is cross, uh, again help of pollination that is required. That is what we call as the xenogamy. Very important, because this leads to the fusion of uh, the gametes from different plants. So they might be genetically different, and since they are genetically different, you see that they have a different ability of adaptation and many more, right? So this is about the different types of uh, you know pollination. Now the next thing that we should learn is about the different types of pollinating agent. Okay, what are the different types of pollinating agents? Pollination by water, insects, and so on. Okay, and that part we will study in our next class, right? So if you have any doubts, please do make a note. We will learn about these things in detail in our next class. See you then. Bye.